Good evening, Pratham. Okay, I'm Rahul Saha, and today we'll be continuing the or our previous discussion on the electron transport chain that we have been talking about. Okay, so electron transport chain, as I had explained to you, contains not only NADH, okay, not only FAD-H2, not only FMNH2, but I had told you that it also contains three more types of electron carriers. Okay, so number one, what I had told you was ubiquinone. Then you have something known as the iron sulfur proteins like cytochrome C, cytochrome B, and cytochrome A. Then you have something known as iron sulfur proteins. Okay, I mean, we have something known as iron sulfur proteins. So what we have is ubiquinone, then iron containing proteins known as cytochrome and iron sulfur proteins. Now, in case of ubiquinone, ubiquinone is used. Why? Because you need to transport the electron from the complex 1 to complex 2, or from the complex 2 to complex 3. So that is the purpose that is solved by ubiquinone. Because the protons just cannot pass through the membrane, so it needs to be pumped via the ubiquinone. And the ubiquinone is a lipid soluble carrier, right? It is a benzoquinone which is lipid soluble. Okay, it is a lipid soluble benzoquinone with a long isoprenoid chain. Okay, so what it becomes is after it takes one electron, okay, after it takes one electron, it becomes a semiquinol radical. One electron and one H plus semiquinol radical, then another H plus, another electron, then it becomes the completely reduced. You can call it fully reduced ubiquinol. And this is your ubiquinone, the ketone group. So we know that the alcohol is the fully reduced version. The you can say aldehyde is the lesser reduced version or more oxidized compared to alcohol. Then you have the ketone, and then the most oxidized version is the carboxylic acid, the COOH group. So from quinone, you're getting quinol means you're getting or you're going towards reduction. Okay. So if I draw the structure for you of ubiquinol, you can just draw the structure. If it's a little bit longer structure. Okay, so what you have is here you have something known as a benzene ring. So on top you have double bond O, double bond O, okay. Then you have CS3, then you have COCS3 this side, then you have COCS3 that side. Then we have a long chain isoprenoid, okay, that is CH2, CH, then double bond C, then CS3, then CH2, and this will be repeated to the power of 10, then H. Now if you add one H plus and one electron to this system, okay, to this molecule, what it will become is, this I can call it as R, okay, it will become one double bond O has become now OH, okay, this has become the radical form O radical, okay, same will stay here, and I can call this the R group to make it this one shorter. This is known as semiquinone. Then if I add another electron to this and H plus, what I'll get is I'll get complete ubiquinol. So both the double bonds have been converted to OH. Now this has become completely reduced ubiquinol. Okay. So this QH2 is containing 2H plus and 2 electrons. So what is this? What are its constituents? It is containing 2 protons and 2 electrons. Like this you can demarcate it. Okay. Now, next is we can go to the next ones. The next ones are, you can say, they are known by the name as cytochromes. Now, you know that cytochromes are the proteins, okay, with characteristic strong absorption of visible light, okay, are the proteins with characteristic, you can say, absorption of visible light. Means they'll be having specific, you can say, their visible, or you can say absorption maximum. Why? Because of their iron containing heme prosthetic groups. Because of their iron containing heme prosthetic groups. Now, mitochondria, they contain three classes of cytochromes, cytochrome A, B, and C, okay, which are designated or distinguished by the differences present in their light absorption spectra only. Okay, each type of cytochrome contains Fe3 plus, and when it accepts the electron, it becomes Fe2 plus. It will reduce and become the ferrous one. Okay, so I'll show you the protoporphyrin ring here. So like that you have protoporphyrin ring in cytochromes also. If you can look at this ring, this is a protoporphyrin ring, and like this you have protoporphyrin ring in case of, you can say, cytochromes 2. This one protoporphyrin ring is attached to one of the polypeptide chain of the hemoglobin molecule, because hemoglobin molecule is composed of alpha 2 beta 2, the adult hemoglobin I'm talking about. Okay, so each chain, each hemoglobin molecule's polypeptide chain, one alpha will be connected to this protoporphyrin ring, beta chain will be connected to this protoporphyrin ring, another alpha will be connected to this protoporphyrin ring, another beta will be connected to this protoporphyrin ring. Now, so four polypeptide chains will be connected to four protoporphyrin rings respectively, independently and individually. Now you have Fe at the plus 2 state here, ferrous state, coordinated by 4n. Here it is connected to the histidine residue of the, you can say, polypeptide chain, which will connect it to the polypeptide chain, and towards this side it will be attached to one, you can say, oxygen molecule. Okay. Now that is why each hemoglobin molecule can contain four oxygen molecules, or can carry four oxygen molecules, or can bind to four oxygen molecules, because the protoporphyrin ring attached to each hemoglobin polypeptide chain can attach to one oxygen molecule. So this is the type of ring which is present in cytochromes also. In cytochrome B, it is the same ring, heme ring, protoporphyrin ring, but in cytochrome C and A, you have heme C and heme A ring, which are a little bit different, okay, to this ring compared to this ring. So if we plot the graph, okay, between wavelength and the percent absorption. Suppose this is 600 nm, this is 800 nm. Okay, visible spectra if we take, okay, so suppose this comes like this. So this is the peak. The peak is at, the peak coincides with the 600 nm one. Okay, So what I can say is this cytochrome, suppose this is cytochrome A, has the maximum absorption at wavelength 600 nm. So cytochrome A has maximum absorption at 600 nm, B has at 560 nm, C has at 550 nm, and you can have the variants also B1, B2, C1. So they will be having different, little bit different absorptive. You can say absorption maximum. Okay. So one more difference that I'll tell you is the heme groups of cytochrome A and B are tightly bound to their associated proteins, but not covalently bound. Okay. So the heme groups of cytochrome A and B are tightly but not covalently bound to their associated proteins. Okay, that is the way it happens. Means the protocovalent ring is attached to the polypeptide chain of cytochrome via van der Waals interaction, dipole-dipole, hydrogen bonding, okay, hydrophobic interaction, any other non-chemical, non-true chemical bond interaction, but not covalent. Whereas the heme group of cytochrome C is bound to the associated protein via covalent interaction. Okay, this is very important because the heme group of cytochrome C binds to the, you can say, polypeptide chain of cytochrome via covalent interaction through its cysteine residue. Cysteine present in the polypeptide chain of cytochrome C, and there are only two amino acids, cysteine and methionine, that comprise of sulfur atom. Okay, otherwise any other amino acids they do not contain sulfur. So they go for disulfide linkage with the amino acid cysteine, okay, which is a covalent bond, right? Now we talk about something known as the iron sulfur proteins, okay, something known as the iron sulfur proteins. Now, iron sulfur proteins, as the name suggests, they also contain iron, okay, but they lack the heme group. So, in iron sulfur proteins, the third category of proteins which can carry electrons and shuttle them from one complex to the other is the iron sulfur proteins which carry iron. Iron is present in them but not in heme. Okay, the iron is not present in heme group, present in heme group. But it is found to be associated with the sulfur atom, which may be the inorganic sulfur atom. It is found to be associated with inorganic sulfur, or it is found to be associated with the sulfur atom of the cysteine residue present in the protein, or both, or found to be associated with the S atom in the cysteine amino acid of the protein, or both these cases can occur. Okay, both these two cases can occur. One of the exceptions to this rule is the very important protein known as the risk iron sulfur proteins, named after the discoverer John S. Riske. You can call them the Riske iron sulfur proteins, RIESKE. Riske iron sulfur proteins, named after the discoverer John S. Riske. Okay. Now, what happens here is, here one Fe atom you can think of is coordinated to two histidine residues rather than two cysteine residues. Okay. So instead of binding to cysteine, the one Fe atom is coordinated to two histidine, another amino acid, two histidine residues, or two histidine amino acid, instead of binding to two cysteine amino acids. So it will bind to the histidine. That's the difference here. Okay, not the cysteine. So these are known as Riske iron sulfur proteins. Okay. So what we have is, we have at least 8 iron sulfur proteins which function in mitochondrial electron transfer chain or electron transport system. Okay, 8 iron sulfur proteins are present. Okay, and if somebody asks you what is the real route of electron flow from NADH to O2, then write this answer. Okay, this is the most correct answer when it comes to the real flow, because I told you that from NADH to complex 1 to complex 2 to complex 3 to complex 4, then to O2, then H2 will be produced. That is fine. That is fine for a layman. But technically speaking, okay, the specific route of electron flow from NADH to O2 will be, I'll put the heading, route of electron flow in electron transport chain. Okay, first will be NADH, then coenzyme Q, okay, then cytochrome B, then cytochrome B1, okay, then cytochrome C, okay, then after cytochrome C it will be cytochrome C1 will come first. Okay, so coenzyme Q, 2B, 2 cytochrome C1 to C, right? Coenzyme Q to B, okay, here I put it, cytochrome C1 to C, okay, NADH to Q to cytochrome B, then C1 to C, okay, then you will be having cytochrome A, then A3, then to O2. Okay, so BC1C AA3, like this you can note it down, BC1C AA3 then to O2. This is the real sequence of transfer of the electron in the electron transport chain, and you have multiple, you can say, compounds that can cause death, okay, of a person by interfering with the electron flow in the electron transport chain. Okay, so number one is known as rotenone. So rotenone is the compound which will impair electron flow, which will obstruct electron flow from NADH to ubiquinone only. And if electron flow is impaired, if it is obstructed, what will happen is we will not be able to generate the electrochemical gradient by the chemiosmotic theory. Thereby, the H pluses will not be becoming high in concentration in the intermembrane space, and then they cannot return via the F1 F0 ATPase or complex 5, and then ATP cannot be generated, and due to lack of ATP, the cell will eventually undergo necrosis or death. Okay. Number two is antimycin. Okay, antimycin A. So antimycin A is cytochrome B to cytochrome C1. That is your antimycin A. It acts here. Okay. Then we have our very definitive killer known as cyanide, or we have also one more thing known as carbon monoxide. So cyanide and carbon monoxide everyone is aware of, and you taste cyanide and you die. Okay, those things are there. What does cyanide do is it impairs the electron flow from cytochrome AA3 to O2. Cyanide or carbon monoxide in high concentration. So you are supposed to have locked in carbon monoxide room filled with carbon monoxide only, you will only survive 5 minutes. Okay. So again, no chemosmotic theory, no electrochemical gradient, no returning of protons and H plus through the complex 5 or F1 F0 ATPase, therefore no formation of ATP and eventual death of the cell and the person. Okay. So these are the, there are various electron transport chain blockers. Okay, these three are the great examples that I gave you. Next we will come to the very first, okay, system, or very first complex, I should be saying. That complex's name is complex 1. Complex 1 is also known by the name as NADH dehydrogenase. Okay, next we come to complex 1, is also known by the name NADH dehydrogenase. Okay. Now, NADH dehydrogenase, why is it important? Okay. The thing is, this is the complex 1, the first, you can say, transmembrane protein that I showed you in the present in the inner mitochondrial membrane, to which NADH will transfer the electrons to. Now, NADH dehydrogenase, okay, this is a huge enzyme, you can say, the integral membrane protein. It acts as an enzyme, and it is a large enzyme, very, very large enzyme, which is composed of, you can say, a gigantic or a huge proportion or a really gigantic number of 45 polypeptide chains. 45 polypeptide chains are present in that enzyme. Okay, just imagine how huge that enzyme is. It contains the flavoproteins, it contains 8 iron sulfur centers also, and the 3D structure, when it folds, it is an L-shaped structure. Okay, now what I'll tell you is, what does this specific complex do, or what does it cause in the form of a reaction that it is so important, so relevant in the electron transport chain. So what it does is it causes the occurrence of, okay, two reactions. One is exothermic, one is endothermic. You can call it thermodynamically, one is exergonic, one is endergonic. Okay, these two are obligatory coupled, simultaneous and obligatory coupled processes because they cannot occur independently. For one to occur, the other must occur. Like that, they are intricately, you can say, related. Number one is the exergonic process. Now, the exergonic process means that it is causing what? It is causing the exergonic transfer of ubiquinone, okay, or exergonic transfer to ubiquinone to, you can say, exergonic transfer to ubiquinone of a hydride ion. Okay, the hydride ion comes from where? It comes from NaDH. Okay, the hydride ion comes from NaDH. Always remember. So I'll write down the reaction for you to easily be able to comprehend it. NaDH plus H plus. Okay, it will attach with the ubiquinone, and ubiquinone will take the electrons in the form of a hydride ion, that is this one, hydride ion. Okay, now the reactions, what will happen is hydride ion, when it has been taken in, or when it has been accepted by the ubiquinone, okay, what you will get is you will get NAD plus QH2. So this has more negative standard reaction potential or more affinity towards the electron, you can say. So it will accept electron more easily. It will have more tendency, more affinity towards electron for its acceptance, and down the gradient acceptance will lead to the energy to be evolved. So this is an exergonic process, and this energy evolved, this energy will be utilized in the next coupled, obligatory coupled process. The next process is, you can say, the endergonic process. The endergonic means the endothermic process. Now the endergonic transfer of four protons from the matrix to the intermembrane space against a transmembrane proton gradient. Okay, the endergonic transfer of four protons means 4H plus from the matrix to the intermembrane space against the gradient, meaning that if you draw it, okay, if this is your complex one, so what is happening is the transfer of electron from NADH, okay, to QH2, energy is evolved, and that is pumping out 4H plus from matrix to intermembrane space, but against the concentration gradient, because H plus concentration is more here compared to H plus concentration in the matrix, so it is going against the concentration gradient. How? It is not utilizing ATP. But the thing is, ATP hydrolysis gives what? Gives energy which can go for uphill transport, just like in the Na plus K plus ATP is. Same way, the energy evolved from this process is used to carry out this process against the concentration gradient for an uphill transport. So 4H pluses are transported every time. This is an endergonic process, but it is possible only due to the energy evolved in the exergonic process. Now 4H plus has been able to be thrown out. Okay, so then 4H plus gets thrown out how? Because the H pluses have been able to be attached to ubiquinol. Ubiquinol will release it. Ubiquinol will also become, you can say, ubiquinone in the process. Okay, so 4H plus will be thrown out. Okay, and you have the production of ubiquinol here. Okay, later on ubiquinol will become ubiquinol. Later on also I'll show it to you in the ubiquinol cycle. So that will happen, and then what we have is the perpetuation of this specific cycle. Now, how is the reduction of ubiquinone coupled to proton pumping? That is the question. Now, reduction of Q occurs far away from the, you can say, membrane arm of the protein where proton pumping occurs. Okay, so the coupling is clearly indirect. Okay, we can think of it as the reduction of Q induces a long range conformational change. Okay, in complex 1. So that happens and that leads to the, you can say, endergonic process to be carried out. Okay, that can happen. Now coming on to complex 2. Complex 2 is known as succinate dehydrogenase. Okay, complex 2 is known as succinate dehydrogenase. Now this is the only membrane bound enzyme which also takes part, okay, in the citric acid cycle, if you remember. Okay, from succinyl CoA to succinate, that is the succinate dehydrogenase. So this is the only membrane bound enzyme that also takes part. Very very important. Very very important 1 mark question, especially for the competitive examinations, which takes part in the TCA cycle also directly. Okay, and it is directly also present in the ETS, so it has dual function. Okay, now complex 2 does what? It couples the oxidation of succinate at one side with the reduction of ubiquinone at the other side. Okay, and those two sides are almost, you can say, 40 angstrom away on the protein. Okay, it is smaller and simpler than complex 1. Still complex 2 contains 5 prosthetic groups, okay, and 2 to 4 different protein subunits. Okay, now they also contain, okay, a heme group like heme B. Okay, and you have the binding site for ubiquinone also. Okay, those things are present. You have iron sulfur centers also, FES centers, we have FAD also. All those things are present in complex 2 as well. Okay, but one thing I'll tell you, keep it in mind that electron transfer, okay, because when I gave you the overview, I gave you a very layman view. Okay, so here we are coming to the technical part of it. So electron transfer through complex 2 is not accompanied by proton pumping across the inner membrane. Very very important, proton pumping will not happen. Okay, so electron, mark this thing, electron transfer by complex 2 is not accompanied by, you can say, proton pumping across the inner membrane. This is a very very important sentence. across the inner membrane, or I can write inner mitochondrial membrane. Okay, inner mitochondrial membrane. Now, although the QS2 produced by succinate oxidation will be then used by complex 3 because electron transport has occurred, so ubiquinone has become a different ubiquinone molecule, has become QH2, but that QH2 production doesn't cause, you can say, proton pumping from matrix to the inner membrane space of the mitochondria, sorry, to the intermembrane space of the mitochondria through complex 2. But that QH2 produced will go and then bind to complex 3, and then it will cause the proton pump through the complex 3 later on, okay, but not through complex 2. Always remember. Now, since complex 2 functions in the citric acid cycle, so factors that affect its activity, okay, they probably serve to coordinate that cycle with mitochondrial electron transfer as well, because it should be available for both the cycles, right? So that is the most important theme here, okay. Now understand very crucially here that the heme compound present of complex 2, okay, is not in the direct path of electron transfer, okay. So what happens is it may serve instead to reduce the frequency with which electrons leak out of the system, okay, moving from succinate to molecular oxygen and produce reactive oxygen species like hydrogen peroxide, like superoxide ion, okay. So these things can happen from complex 2, okay, H2O2 production, superoxide ion, okay, this is superoxide ion. So there are very few individuals who have point mutations in complex 2, okay, near the heme B or the ubiquinone binding site. They suffer from a great disease, okay, known as hereditary paraganglioma. This characterizes the benign tumors of the head and neck, commonly in the carotid body, which I'll be talking about later on in the respiratory tract when I'll be teaching you that chapter. That is responsible for sensing, you can say, O2 level in the body, okay. So what happens is you can directly go for, okay, you can directly go for oxidative damage, and its, you can say, cure also is present in the mitochondrial transport, you can say, electron transport chain itself, okay, the cure is present, okay. So what happens is what you have is you have excessive oxidation occurring. It can occur through the complex 2, okay, and when that occurs through complex 2, you can produce H2O2, you can produce O2 minus ions, you can also produce something known as hydroxyl ion, OH minus, okay. So those things can be produced. So several steps in the path of oxygen reduction in mitochondria have the potential to produce highly reactive free radicals that can damage cells. Now, one thing I'll tell you, the passage of electrons from QH2 to complex 3, once QH2 has been produced, via complex 2, it will not pump protons out of the inner mitochondrial membrane, it will move to complex 3. And the passage of electrons from complex 1 to 2 to QH2 involve the radicals Q minus, right? That is known as the semiquinol radical, okay, Q minus. As an intermediate, the Q minus radical can, with low probability, pass the electron to O2, okay. Instead of holding it, it can pass the electron to O2, making it a superoxide, you can say, free radical, because you have Q to this one, right? This one you have already. Now this is present between it becoming QH2 and Q. So this is the midway, okay, from complex 1 and 2 to QH2. So there is a probability that it will not be like this and wait for another one and become QH2. It may in turn transfer its electron, okay, to oxygen. So O2, it will become O2 minus superoxide radical. Superoxide radical. Now, when superoxide radical has been produced, you are in a big problem. Okay. Now, its formation also leads to production of even more reactive hydroxyl free radical like OH radical. That can happen. Okay. So this is even worse, OH radical. Okay. Now, reactive oxygen is species. Okay, that's why antioxidants are taken in the food. Reactive oxygen species like this one can wreak havoc. It will react with and damage enzymes, membrane lipids, nucleic acids. Okay. So in actively respiring mitochondria, 0.2% to 2% of the O2 used in respiration forms this superoxide radical. Okay, which is more than enough to have lethal effects unless the free radical is broken down. It is quickly disposed of. It is quickly quenched. So what quenches this one. Now the formation of ROS is favored when two conditions are met. Mitochondria are not making ATP, okay, and therefore have a large proton motive force, okay. So when they have a large proton motive force, meaning that if they have a large ratio of QH2 to Q, more QH2 will not be, you can say the production of more QH2 will not be favorable. So when then the Q will be converted to Q radical, it will not become this, it will transfer it to oxygen and make this one. Okay. So then the mitochondria is not making that high amount of ATP at a very high rate. This will develop, high ratio will happen. Okay. This ratio will become high. Then factor number two that comes into existence, which favors the formation of free radicals, is there is a high NADH to NAD plus ratio in the matrix. Okay. Now again, this will keep on causing, you can say, flow of electrons. Okay, and more electrons are available to enter the electron, the respiratory chain, that can be immediately passed. Okay, then can be immediately passed through to oxygen. Okay, means if you have more NADH to NAD plus in the matrix, again that will lead to, you can say, more and more formation of QH2 in the end, and that will increase the QH2 to Q ratio, and that will again cause the, you can say, balance to tip towards the formation of superoxide radical. Okay, so these things, they favor the, you can say, formation of free radicals. Okay, so now the free radical must be quenched. Now to prevent oxidative damage by O2 minus, cells have the enzyme known as superoxide dismutase. Okay, what it does is it catalyzes the following reaction. Okay, so now 2O2 minus plus, if you go for 2H plus, what will happen is you will get H2O2 plus O2. So this is catalyzed by superoxide dismutase. So it is converting this O2 into H2O2. Okay. Now the the hydrogen peroxide produced will be rendered harmless later on by the another enzyme known as glutathione peroxidase. Okay. Glutathione peroxidase will render it completely, you can say, ineffective. It will convert it into H2O. Okay. So what will happen is, then you can think of the, you can say, conversion of H2O2 to H2O via glutathione peroxidase. So how the glutathione peroxidase works, okay, and the Q cycle will be explained in the next class. So I hope you are able to comprehend this lecture. If you had any doubts, you can contact me on my personal number. So we will be continuing our discussion in the next class as well. Okay, then thanks a lot. Let's